Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Seeking and its connection to all jawed vertebrates. Ready? Let's go! Jawed vertebrates, sometimes also referred to as gnathostomes, are a systematic group within the subphylum of vertebrates. This taxon includes all currently living extant vertebrates, except for the primitive lampreys and hagfishes, which comprise about 100 species. In total, jawed vertebrates encompass over 54,000 species, including cartilaginous and bony fishes, amphibians, reptiles, birds, and mammals, 99.8% of all vertebrate species, including humans. Jawed vertebrates first evolved during the Paleozoic era, with their fossil record beginning in the early Silurian period with the acanthodians, spiny sharks. Since the Middle Devonian, approximately 380 million years ago, they have represented the majority of vertebrate life. The most important autapomorphy, distinctive derived trait, of jawed vertebrates is the reinforcement of the mouth margins through articulated cartilage or bony arches. This led to the development of a typically toothed jaw, enabling these animals to grasp, hold, and break down food, opening up entirely new dietary possibilities. According to the classical model of jaw evolution, the mandibular arch, jaw arch, was derived from a gill arch, a skeletal support structure located between the gill pouches. However, it remains uncertain which specific gill arch was involved. One hypothesis suggests it was the third gill arch, with the two anterior premandibular arches having regressed. Cartilage structures in the skulls of Neoselacians, modern sharks and rays, known as labial cartilages, may be remnants of these reduced anterior gill arches. The jaw arch in gnathostomes consists only of the middle two arch elements, which are connected by the primary jaw joint, or in mammals by the secondary jaw joint. Powerful adductor muscles serve to close the jaws. The gill arch that follows the jaw arch becomes the hyoid arch, also called the hyale or hyoid arch, while the remaining gill arches, typically 5, with few exceptions, retain their basic structure in gnathostomes as supports for the gill apparatus. Each gill arch consists of 4 skeletal rods: pharyngobranchial, epibranchial, ceratobranchial, and hypobranchial. Between the jaw arch and the hyoid arch lies a spiracle in the basic gnathostome blueprint, through which respiratory water can be drawn in. Teeth evolved from dermal bone scales and may be located along the jaw margins, within the oral cavity, or in the pharynx. An alternative hypothesis suggests that jaws are homologous to cartilaginous structures found in the velum of lamprey larvae, amocoete larvae. The velum is located in the pharynx of these filter-feeding organisms and, together with the gill musculature, generates the water current from the mouth opening to the gills. According to this hypothesis, jaws evolved from a structure that had always served a role in feeding. The axial skeleton initially consists of the notochord, chorda dorsalis. Vertebrae developed multiple times independently to reinforce the notochord and are therefore not considered autapomorphies of jawed vertebrates. Among the unpaired fins, the dorsal fin and caudal fin were inherited from jawless ancestors, while the anal fin is a new development that did not appear in the most primitive gnathostomes, the placodermi. The original caudal fin was heterocercal, meaning the end of the vertebral column bends upward and supports the larger upper lobe of the tail fin. Another key autapomorphy is the presence of paired pectoral and pelvic fins supported by skeletal elements. These fins significantly improved the maneuverability of gnathostomes and eventually gave rise to the forelimbs and hind limbs of terrestrial vertebrates. The extinct jawless osteostraci also had paired pectoral fins, but these lacked skeletal support and were held in place only by muscles. The pectoral fins are articulated via the shoulder girdle, and the pelvic fins via the pelvic girdle. These paired fins are moved by muscle groups known as elevators, lifters, extensors, and depressors, lowerers, flexors. The eyes of jawed vertebrates possess a true cornea. The position and innervation of the 6 external eye muscles differ from those in jawless vertebrates. The development of internal eye muscles allows the eye to focus objects at varying distances, from near to far, onto the fovea of the retina in both eyes, enabling accommodation and sharp vision. As olfactory organs, 2 nostrils develop. The balance organ acquires 3 semicircular canals, one for each plane in 3-dimensional space. A lateral line system and electroreceptors are also likely part of the basic blueprint of jawed vertebrates. The brain of jawed vertebrates is divided into 3 regions: the prosencephalon, forebrain, mesencephalon, midbrain, and rhombencephalon, hindbrain. From the neural tube, a spinal nerve emerges for each muscle segment, which splits into a dorsal and a ventral branch. Glial cells along the axons of peripheral nerves enable rapid transmission of information. In the digestive tract, a stomach evolved as a storage organ. In primitive gnathostomes, the surface area of the intestine is increased by a spiral valve. The liver and pancreas are the largest digestive glands and originated from the endoderm. The digestive tract, kidneys, and reproductive ducts, egg and sperm ducts, all terminate in a cloaca. Jawed vertebrates form a taxon within the subphylum Vertebrata. Among all jawless vertebrates, the extinct osteostraci, possibly together with the Paturiaspida, share the greatest number of autapomorphies with jawed vertebrates, especially paired muscular pectoral fins, a bony ring, scleral ring, around the eyes, and the heterocercal tail fin. They may represent the sister group to the jawed vertebrates. Cypriniforms, commonly known as carps, and their relatives, are a highly diverse order of freshwater fishes. They are found across Europe, Asia, Africa, and North America, and often dominate the local fish fauna. About 1/3 of all freshwater fish species and 6% of all vertebrate species belong to this group. The greatest species diversity occurs in Southeast Asia. Cypriniforms prefer warmer waters and are increasingly replaced by trout species in colder northern regions and mountain streams. They are absent from South America, Madagascar, Australia, and regions east of the Wallace Line. Among the most well-known members of this group are the common carp, Cyprinus carpio, and the zebrafish, Danio rurio, which is widely studied as a model organism in biological research. Cypriniforms are typically slender to deep-bodied fishes. Within the loach family, there are also eel-like elongated forms, such as the weather loach and spiny loaches. Species from temperate regions and larger tropical forms tend to be plain in appearance, while smaller tropical species are often brightly colored and popular in aquariums. The jaws, other oral bones, and gill arches 1 through 4 are toothless. Instead, 1 to 3 rows of pharyngeal teeth are located in the throat, mounted on sickle-shaped pharyngeal bones. These teeth are an important feature for species identification. The mouth is often highly protrusible, and the lips are frequently equipped with barbels. A bone called the kinethmoid, located between the processes of the premaxilla, is characteristic of cypriniforms. The head and gill covers are naked, while the body is covered with elasmoid scales. A fat fin is absent. Intermuscular bones, ossified connective tissue between muscle segments, are often numerous. The swim bladder is divided into 2 or 3 chambers by constrictions and is connected to the foregut, though it lacks an internal wall. Most cypriniforms are small to medium-sized fishes. The largest is Tor putatora, a South Asian cyprinid that can reach a length of 2.75 meters. Interestingly, the smallest known fish is also a member of this group, Paedocypris progenetica, which reaches a length of just 10 millimeters. Most cypriniforms are small to medium-sized fish. The largest is Tor putatora, a South Asian cyprinid that can reach a length of 2.75 meters. Interestingly, the smallest known fish is also a cypriniform, Paedocypris progenetica, which reaches just 10 millimeters in length. The order Cypriniforms, along with Characiforms, characins, Siluriforms, catfishes, and 2 other less species-rich families, belongs to the superorder Asteriophysi, which includes the majority of all freshwater fish. Cypriniforms comprise 23 families and around 4,300 described species. The number of yet undescribed species is estimated at about 2,500. Many of the families currently listed were previously considered subfamilies of Cyprinidae and were only elevated to family status in 2016 by Stout and colleagues. Cypriniforms include the most primitive of the Asteriophysi in the narrow sense, i.e., excluding the Gonorhynchiforms. This is evidenced not only by physiological details, but also by their great distribution, which indicates they had the longest time to spread. The earliest that cypriniforms might have diverged from Characifici, Characiforms and relatives, is thought to be about the early Triassic, about 250 million years ago, Maya. 22. However, their divergence probably occurred only with the splitting up of Pangaea in the Jurassic, maybe 160 million years ago, Maya. By 110 Maya, the plate tectonics evidence indicates that the Laurasian cypriniforms must have been distinct from their Gondwanan relatives, 23. The cypriniforms are thought to have originated in Southeast Asia, where the most diversity of this group is found today. The alternative hypothesis is that they began in South America, similar to the other otophycans. If this were the case, they would have spread to Asia through Africa or North America before the continent split up, for these are purely freshwater fishes. As the Characiforms began to diversify and spread, they may have outcompeted South American basal cypriniforms in Africa, where more advanced cypriniforms survive and coexist with Characiforms. 24. Until 2025, no cypriniform fossil remains were known predating the Cenozoic. In 2025, multiple fossil remains of an indeterminate cypriniform, including jaws, tooth plates, and vertebrae, were identified from the early Maastrichtian aged, 71 to 69 Maya, Prince Creek Formation of Alaska. This occurrence suggests that in contrast to previous hypotheses, cypriniforms may have originated in high-latitude regions and radiated southwards during the Cenozoic. The next oldest cypriniform fossils are already assignable to the living family Ctostamidae. From the Paleocene-aged Paskapoo Formation of Alberta, they are roughly 60 million years old. During the Eocene, 55 to 35 Maya, catostamids and cyprinids spread throughout Asia. The earliest members of the cyprinid subfamilies, Barbinae and Danioninae, are known from the Eocene Sangkarawang Formation of Indonesia, in addition to possibly Smilogastrinae and Labioninae. The extinct family Jianghenicthidae is known from the Eocene of China. In the Oligocene, around 30 Maya, advanced cyprinids began to outcompete catostamids wherever they were sympatric, causing a decline of the suckers. Cyprinids reached North America and Europe about the same time, and Africa in the early Miocene, some 23 to 20 Maya. The cypriniforms spread to North America through the Bering Land Bridge, which formed and disappeared again several times during the many millions of years of cypriniform evolution. The Cyprinidae, carps and minnows, are the largest family within cypriniforms. This family includes well-known freshwater fish such as the common carp, Crucian carp, Prussian carp and its domesticated form, the goldfish, as well as many small species popular in aquariums. It also includes the largest cyprinid species in the world, such as tortor from the Indian subcontinent and the giant barb, Catlocarpio siamensis from Southeast Asia. With over 1,300 species, Cyprinidae are the most species-rich family of cypriniforms, which total around 4,300 species. Including about 400 closely related species, they contain more polyploid species than any other fish group, approximately 100 tetraploid and 150 hexaploid species. These likely originated through hybridization between different ancestral species, followed by rapid adaptive radiation. Most cyprinid species are found in southern Eurasia and Africa, with others occurring in northern and central Europe or Central Asia. The common carp and other species important for human consumption have been introduced worldwide and now inhabit waters in regions that were previously carp-free, such as South America, Madagascar, Australia, and New Zealand. The Cyprinidae family includes both very small species, as little as 3 centimeters, and very large ones, up to 2 meters. Their bodies range from elongated to deep-bodied and are only slightly compressed laterally. The mouth is either terminal or subterminal, and the lips are thick and fleshy, often adorned with structures like tubercles, papillae, or fringe-like projections. Barbels may be absent or present in 1 or 2 pairs. The jaws are toothless, but pharyngeal teeth are fused to the pharyngeal bones and are used for species identification. These teeth are arranged in 1, 2, or 3 rows, and typically press against a horny plate on the underside of the skull, known as the carpstone or grinding plate. The mouth structure is characteristic of each genus and serves as an important taxonomic feature. Cypriniforms lack a true stomach. The esophagus leads directly into the midgut. The swim bladder is divided into 2 or more chambers and is connected to the inner ear via a series of 8 small bones known as the Weberian apparatus, which enables efficient sound transmission. Other distinctive features of cyprinids include the spawning rash and the production of a fright substance, alarm pheromone in specialized skin cells, which alerts schooling fish when one of them is attacked by a predator, such as a pike or heron. The dorsal fin is usually located directly above the pelvic fins. The first fin rays of the dorsal and anal fins are ossified and unbranched, and the rear edge of the first ray may be serrated. The short anal fin typically has 5 to 7 branched soft rays. That's it for now. In the next episode, we take a closer look at Starmie and its surprising connection to all Deuterostomia Pokemon. Feel free to subscribe to our channel, leave a like, and check in next time.